Good afternoon, dear students. Today we'll be discussing about histologic structure of two organs of the digestive tract called an esophagus and a stomach. The esophagus is the upper part of the digestive tract, connecting the oral cavity to the stomach. The major function of the esophagus is to provide passage for food from the mouth to the stomach. The length of the human's esophagus is about 25 centimeters. This organ can be divided into three regions: the upper, middle, and lower esophagus. Like other parts of the digestive tract, the esophagus has four tunics in its wall. They are mucosa, submucosa, muscularis externa, and adventitia or serosa. The innermost mucosa of esophagus consists of three layers. They are epithelium, lamina propria, and muscularis mucosae. The mucosal epithelium, lining the lumen of esophagus, is non-keratinized stratified squamous. It's developed from the precaudal plate for embryogenesis. The lamina propria of esophageal mucosa is loose connective tissue. The lamina propria of the upper and especially lower esophagus contains some tubular alveolar mucous glands called esophageal cardiac glands because they are similar to the cardiac glands of the stomach. The muscularis mucosae is absent or poorly developed in the upper esophagus. It appears only at the level of the cricoid cartilage of the larynx and becomes thickest in the lower esophagus. The muscularis mucosae is composed of longitudinally organized smooth muscle cells. The submucosa of esophagus is loose connective tissue, containing a lot of mucous glands, secretory parts, throughout esophagus length. The submucosal glands are called esophageal glands proper. Structurally, they are compound, branched, tubular alveolar glands, primarily producing mucus to lubricate the esophagus inside. The muscularis externa of esophagus consists of two muscle layers, an inner circular and an outer longitudinal. There are various types of muscle tissue in the different regions of esophagus. Striated muscle tissue there is in its upper part. Both striated and smooth muscles there are in its middle portion, and only smooth muscle tissue there is in the lower part of esophagus. The outermost tunic of esophagus is commonly covered by a layer of loose connective tissue called the adventitia, which blends into the surrounding tissue. Only the lower esophagus in the peritoneal cavity is covered by serosa. Serosa is represented by loose connective tissue and a simple squamous epithelium called mesothelium. This is the photomicrograph of the esophagus in cross-section. The lumen of esophagus has an irregular shape because two inner tunics of this organ, the mucosa and the submucosa, produce several longitudinal folds into the lumen of this organ. Next, our question is called stomach. The stomach is a dilated segment of the digestive tract starting its middle part. The stomach temporarily stores food, mixes food with gastric juice, and initiates digestion of food. The gastric juice produced in the stomach is a mixture of the hydrochloric acid, pepsin, mucus, intrinsic factor, water, and electrolytes. In stomach anatomically there are four regions called cardia, fundus, body, and pylorus. But histologically there are only three regions, because the fundus and the body of stomach are identical in microscopic structure. Thus, the histologic regions of stomach are the following: cardiac region, which is the part near the esophageal orifice; pyloric region, which is the part proximal to the pyloric sphincter; and fundic region, which is the largest part of the stomach situated between the cardia and pylorus. Like other parts of the digestive tract, the wall of stomach has four tunics. They are mucosa, submucosa, muscularis externa, and serosa. The innermost mucosa of stomach consists of epithelium, lamina propria, and muscularis mucosa. Third tunic of stomach, called muscularis externa, is very thick in this organ. It consists of three layers of smooth muscle tissue: an inner with oblique arranged smooth muscle cells, a middle with circular arranged smooth muscle cells, and an outer with longitudinal arranged smooth muscle cells. The gastric mucosa is covered by a simple columnar epithelium that invaginates into the lamina propria, forming the gastric pits. The gastric epithelium is originated from interderm for embryogenesis. All cells of this epithelium secrete mucus and are called mucous surface cells. Mucus protects the gastric mucosa against acid and pepsin secreted by the gastric glands. The gastric glands within the loose connective tissue of the lamina propria open into the bottom of the gastric pits. The muscularis mucosae of the stomach mucosa is well developed. It's a layer of smooth muscle composed of an outer longitudinal and an inner circular layers. The submucosa of stomach is composed of loose connective tissue and blood and lymph vessels, and is infiltrated by lymphoid cells and mast cells. In stomach, the mucosa and submucosa together produce the longitudinal folds or ridges called rugae. In lamina propria of the mucosa there are three types of gastric glands. They are cardiac glands lying near the esophageal orifice, pyloric glands lying in the pyloric antrum, and fundic or main gastric glands that are represented in fundus and body of the stomach. The most numerous fundic glands produce the principal components of the gastric juice. Structurally, they are simple, branched or unbranched tubular glands that extend from the bottom of the gastric pits to the muscularis mucosae. The fundic glands can be divided into three parts, called isthmus, neck, and base or fundus. There are five types of cells in fundic glands. The cells are the following: mucous neck cells, chief cells or zymogenic cells, parietal cells or oxyntic cells, undifferentiated cells, and interendocrine cells. Mucous neck cells localize in neck region of the fundic gland. They produce mucus. Chief cells localize in fundus of the fundic gland and produce two types of enzymes, pepsinogen and lipase. Parietal cells are distributed throughout the fundic gland. They produce hydrochloric acid and an intrinsic factor. Undifferentiated cells localize in isthmus of the gland. These cells are stem cells having capacity to mitotic activity to provide the regeneration of the surface epithelium of stomach and glands epithelium. Interendocrine cells localize in neck region and fundus of the gland. They produce different hormones and hormone-like regulating agents such as gastrin, serotonin, histamine, and somatostatin. The most numerous cells of stomach are chief cells and parietal cells. Chief cells are typical protein-secreting cells. The abundant rough endoplasmic reticulum in their basal cytoplasm gives this part a basophilic appearance, whereas the apical cytoplasm is eosinophilic owing to the presence of the secretory vesicles called zymogen granules. Zymogens are inactive enzymes that are precursors of active forms of enzymes. Chief cells secrete pepsinogen and weak lipase. On contact with the acid gastric juice, pepsinogen is converted to a proteolytic enzyme called pepsin. Parietal cells are triangular cells, sometimes binucleate, and appear as eosinophilic cytoplasm. The parietal cells contain a lot of mitochondria and extensive intracellular canaliculi that communicate with the lumen of the gland. Each intracellular canaliculus of cells is a deep invagination of its apical plasma membrane with numerous microvilli. Parietal cells produce the hydrochloric acid presented in gastric juice. This process takes place interparietal cell intracellular canaliculi, where both hydrogen ions and chloride ions are actively transported across the cell membrane. In stomach, the hydrochloric acid creates an acidic environment to produce active enzyme pepsin from precursor pepsinogen. Parietal cells also produce an intrinsic factor, which is required for the absorption of B12 vitamin. This is the photomicrograph of the stomach fundus and fundy glands presenting in its lamina propria. Two types of cells are well recognizable in fundy glands. They are singly line axiphilic cholestanian parietal cells and groups of basophilic cholestanian chief cells. The pyloric region of stomach has several differences from the other portions of this organ. These differences are the following. Deep gastric pits that occupy about half the thickness of the mucosa, greatly thickened the middle circular layer of muscularis externa to produce the pyloric sphincter, and pyloric glands in the lamina propria. Pyloric glands are branched, coiled tubular glands. They are more short, with a more wide lumen than fundy glands, and contain primarily mucus-secreting cells and also stem cells and two types of enteroendocrine cells, gastrin-secreting or G cells and somatostatin-secreting cells or D cells. The mucus secretion of pyloric glands protects the epithelium of the pylorus from hydrochloric acid actin. Enteroendocrine G cells release gastrin, which stimulates parietal cells to secrete hydrochloric acid. Enteroendocrine D cells release somatostatin, which inhibits the release of gastrin by G cells. The glands in the lamina propria of the cardia are called cardiac glands of stomach. Structurally, they are branched tubular glands with coiled secretory portion. The cardiac glands contains mainly mucus-secreting cells and also enteroendocrine cells, stem cells, and a few parietal cells. The mucus-secreting cells produce mucus and lysozymes. The mucus protects the stomach wall from acidic gastric juice. The lysozymes are bacteriostatic agents which destroy bacterial membrane, preventing bacterial infections. It was last question of this presentation. Thank you very much for your attention.